right. Well, we're just going to wait a few minutes and let a few people join that are coming in and clicking the links, but we'll get started here shortly with our vet chat of first one of 2021. All right. Um, First, I'd just like to say thanks, Dr. Frampton, for your time today. Uh, we tried to schedule this last week. I had a scheduling error, a user error on my part. So Dr. Frampton was kind enough to reschedule this before he attempts to start teaching tomorrow at the University of North Carolina, Wilmington. This is a great vet chat scheduled for this week, and it's going to be about equine herpes virus 1. As we know, this is a very contagious virus for horses, and left untreated can be deadly. The symptoms, signs, and everything Dr. Frampton will talk about and what his research is doing and what they're doing at the University of North Carolina, Wilmington on some projects that Grayson has actually funded. So without further ado, I'll shut my microphone and video off now and turn it over to Dr. Frampton. Again, thanks so much for your time. Thank you for your knowledge and your passion that helps us move forward with the horses. So thanks, and I'll turn it over now.
Okay, thanks, Jamie. And I want to begin by thanking Grayson for giving me the opportunity to talk a little bit about the research that we've done over the past, 15 or 20 years. And a lot of this research that I'll talk about today, as Jamie said, has been funded by the Grayson Jockey Club. So I'm really thankful to have this opportunity.
Let me jump in real quick too. For those participating on the webinar live, you can ask questions or your Q&A button at the bottom of the Zoom software, and we'll save all those questions to the end. So as Dr. Frampton says, ask him about EHV.
Yeah, that's a mug that one of my former students got me as a parting gift. so
here's your chance, and just use the Q&A button at the bottom. Thanks. I'll disappear.
Great, cool, thanks. All right, so I guess we'll just go ahead and jump on in. I'm going to talk about two main areas of research today. First of all, we'll talk about how we identified an entry receptor for EHV. A lot of you guys probably have heard in the news a lot about viruses lately, particularly SARS-CoV-2, which is the caused invasion of COVID-19. Just like that virus needs a receptor to bind to a receptor to get into cells, all viruses, including EHV, need to bind to a receptor. So I'll talk about how we identified a receptor for EHV-1. That'll be the first part of the talk. And then I'll talk about some of our current Grayson-funded research, and that is the investigation of different anti-inflammatories for their ability to reduce EHV replication, with the idea being that if we can reduce the amount of virus that's present in the horse, we might go a long way towards limiting or reducing the sequelae that we see, including abortions and the neurologic disease, equine herpes virus myeloencephalopathy. So this slide here shows the lab where we do most of our work. I was telling Jamie that this was a picture taken before Hurricane Florence in 2018. Hurricane Florence blew into Wilmington, blew a hole in the roof of Dobo, and this building was basically torn down to the studs and rebuilt, and we just got back in here in August. Fortunately, we found another lab to continue our research across campus, but it's nice to be back. But here's just the general setup of the lab. We've got our benches here. We've got a couple of biosafety cabinets where we do all of our tissue culture work. Here's one of my former students, Isabel, doing some work. All right, so here we go. EHV, equine herpes virus type 1, is an alpha herpes virus. It's in the same family as chickenpox viruses and the herpes simplex viruses. It's enveloped. Here's a schematic that you'll see again and again throughout the talk. This was a diagram or a picture of the virus that a buddy of mine at the University of Pittsburgh put together. So I use this to represent a equine herpes virus particle, or what we call a virion. Here's an actual electron micrograph image that I took of, so this is a part of a cell that's been magnified many thousands of times. And here we've captured an equine herpes virus particle attaching to the surface of the cell. So a lot of our work initially was aimed at trying to better understand how this virus interacts with the cell. How does it bind to the cell? And then how does it get inside of the cell? And then ultimately, how does it make more copies of itself? So EHV, as I said, is an alpha herpes virus. It's enveloped, so you have this nice lipid envelope on the surface of the virion, and protruding from the envelope, you have 12 separate glycoproteins that carry out different roles for the virus. And I'll talk about a few of these glycoproteins and what they do for the virus in a minute. The tan region right here, right below the lipid envelope, between the envelope and the protein capsid, is a region that's been termed the tegument. And this region has some key viral enzymes that the virus brings in with it that help the virus replicate. It has what we call an icosahedral capsid, this 20-sided die-shaped capsid. This is the protein capsid that encloses the double-stranded viral DNA. And the DNA genome is fairly large as far as viruses go. It has about 150,000 base pairs, and 76 genes are encoded within the virus genome. In terms of EHV pathogenesis, or the diseases that are caused by EHV, most horses are infected very early in life. They have a respiratory tract infection. They have symptoms similar to a common cold. They may have fever, malaise, loss of appetite, some discharge as shown here. But most horses recover from the respiratory infection within about 1 to 2 weeks, and then they're fine for the most part. However, the virus remains inside of the horse, right? Just like all herpes viruses, EHV establishes latency within the horse, so it's with the horse for the horse's entire life. And this virus that's latent can reactivate periodically and cause disease again, and also be shed to other horses to infect them. A couple of sequelae that can occur post-infection include abortogenic disease and neurologic disease. Both of these occur subsequent to the virus being spread in the bloodstream in what's called a viremia. So you have viremia, the virus gets into the bloodstream and then infects endothelial cells within the uterine cavity or endothelial cells within the central nervous system. This causes thrombosis, hypoxia, ischemia. You get this inflammation that ensues, and this can lead to expulsion of a fetus for abortions. And for the neurologic disease, equine herpes virus myeloencephalopathy, this can lead to paralysis and also, and in some cases, horses have to be put down. So these are two sequelae that we really want to better understand so that we can come up with some therapies to prevent these conditions. And importantly, while there are vaccines on the market, none have been shown to be effective in blocking the neurologic disease. So I think there's still a lot of work that has to be done in terms of drugs, antiviral drug development, as well as coming up with better vaccines. So this is a slide that I took from one of my colleagues, Klaus Osterreiter. He put this together, and it does a good job of basically showing what happens, where the virus goes after a horse has been infected. So let's say a horse is infected. This is a primary infection. The virus is breathed in by the horse. It sets up an infection in the upper respiratory tract. You have these respiratory epithelial cells that are infected with the virus. The virus replicates in these respiratory epithelial cells. The virus then makes its way to draining lymph nodes where you have further replication. And then ultimately, you have these peripheral blood monocytes that are going to carry the virus to other locations in the horse in what's termed a cell-associated viremia. So the virus actually hitches a ride from the respiratory epithelium. It gets into these peripheral, and these PBMCs now carry the virus to other tissues within the horse. And one target cell type are endothelial cells. These endothelial cells within the central nervous system or in the pregnant uterus can become damaged. You have inflammation that comes, you have lymphocytes that are recruited and other inflammatory mediators and immune cells come into this region. And if you get severe inflammation, this can lead to these two sequelae. So anything that we can do, and this is something that was shown years ago by George Allen and others, anything you can do to reduce the amount of virus that's present in this viremia or present in the bloodstream, they're going to go a long way towards preventing these two disease outcomes. So that's part of what I'll talk about today. We've got two main cell types that we work with in the lab. We've got epithelial cells that we look at to help us gain a better understanding of how the virus replicates and interacts with these epithelial cells. And then we've got some equine endothelial cells that my colleague Udini Balasaria gave to me a while ago. And we infect these cells and look at the consequence of infection in these cells. So we've got a couple of good cell lines available in the lab to do our work to see how this virus interacts with these different cell types. And then of course, as I mentioned earlier, once the virus infects a horse, the horses remain infected for their entire life. The virus establishes what we call latency and can reactivate, and this cycle can repeat. Okay, so a little bit about the EHV life cycle and kind of what we knew at the beginning of doing our work with EHV, and then I'll tell you, we'll walk through some of the data, and then we'll see where we are, what we learned over the past 10 or 15 years. So you can imagine that this is a cell here that's going to be infected with EHV. So here's the virion. We know that the virus has to attach to the cell surface. Here's the plasma membrane of the cell. And at the time we started our work, there was no identified cell entry receptor for EHV. So what the virus binds to in order to get into the cell was not known at the time. So I'm going to talk about the research that we did to figure out or to identify one of these cell receptors. So after the virus binds to a receptor, the virus then has to traverse this fairly formidable membrane, this phospholipid bilayer that's present that makes up the plasma membrane. So the virus has to get across this plasma membrane to get inside of the cell, and then it has to get its DNA down into the nucleus so that new virus particles can be generated. So we did a lot of work, and we published some papers detailing this process. I'm not going to talk about that in today's talk, but we have shown that this virus actually utilizes cellular microtubules to make its way from the periphery of the cell down to a nuclear pore where the viral DNA is injected into the nucleus so that virus replication and new particle generation can ensue. So if you want to read more about that, we published that back in 2007 and 2010. Once new virus particles are generated in the nucleus, this is where the new capsids are formed and the double-stranded DNA. The new viral DNA is packaged in these capsids. They then have to make their way back and outside of the cell. So they go through this maturation process, and they ultimately obtain their final envelope in the trans-Golgi network and then are released from the cell. And this is a lytic virus. So during infection, as the infection is wrapping up and the life cycle is complete, the virus is going to lyse or kill these cells, and then all of these virus particles that are generated are going to be released and can infect new cells. So as I said, we were very interested in looking at this the real early events of EHV, the EHV life cycle, and that is, what does it bind to, right, on the cell surface in order to allow it to get inside of the cell? At the time, there was some information regarding attachment of the virus to cells. This was work done by Sugihara's group and Osterrieder's group. where they showed that these two glycoproteins, glycoprotein B and glycoprotein C, these glycoproteins on the surface of the virus, actually bind to these sugar molecules called glycosaminoglycans on the cell surface. So GB and GC, you can think of these guys as kind of the glue binding the virus to the cell surface. However, it was known that this binding mediated by GB and GC was not sufficient for entry of the virus into the cell. So you needed a second event to occur. And we and other groups knew at the time that GD was also required for entry, because if you knocked this glycoprotein out of the virus was unable to enter cells. So this is kind of what we knew at the beginning of this process. But what we didn't know, again, was what is this entry receptor that's present on the cell surface that EHV has to bind to in order to get inside of the cell. All right, so at the time I started this work, I was doing a postdoc at the University of Pittsburgh School of Medicine in the lab of Joe Glorioso. And he was gracious enough to let me continue equine herpes virus work. Of course, as long as I was contributing to the lab, writing grants and so forth. And so this is where we started to investigate and to try to identify the entry receptor. And at the time, there were entry receptors that had been identified for related alpha herpes viruses. So we initially tested to see whether EHV, since it's an alpha herpes virus, could utilize any of the previously identified entry receptors. And two of those are shown here. We've got the first one, which was originally called HVEM for herpes virus entry mediator. This was identified by Rebecca Montgomery back in 1996. She got a nice cell paper out of it. This is a TNF receptor superfamily member. It's now been renamed Heavy A because Heavy B and Heavy C were identified. And so this one here was the first one identified to enable or to be used by an alpha herpes virus to gain entry into a cell. And then Rob Garrity and his group in 1998 came along and identified Nectin-1, also called Heavy C, as another receptor, entry receptor for alpha herpes viruses. And actually this one now is known as the primary receptor for alpha herpes viruses such as herpes simplex virus type 1 and 2. And this is an immunoglobulin superfamily receptor. So we had in Joe's lab at the University of Pittsburgh, fortunately, he had cell lines already that expressed this receptor and this receptor. And so the idea was to infect these cells with EHV to see whether or not EHV could utilize either one of these receptors to gain entry into the cell. So here's the cell lines that we had at our disposal to use. These are some Chinese hamster ovary cells. CHO-K1 cells don't have Heavy A or Heavy C. So these cells are highly resistant to these alpha herpes viruses, HSV-1, HSV-2, and bovine herpes virus. If you infect CHO-K cells with these viruses, the cells cannot be infected. However, if you introduce Heavy A or Heavy C and you make these Chinese hamster ovary cells now express these receptors on their surface, and then you infect with these herpes viruses, this is what you get. You see that HSV-1 and HSV-2 can productively infect cells now that express Heavy A. Bovine herpes virus couldn't infect through Heavy A. And if you look at Chinese hamster ovary cells expressing Heavy C, now each of these alpha herpes viruses can productively infect these cells. So what this told us was that Heavy A and Heavy C are indeed bona fide entry receptors for these alpha herpes viruses. The question we had was, does EHV utilize either Heavy A or Heavy C for entry into cells? So that's what we did first. And so I'll start here by telling you that when I was in grad school working for Dennis O'Callaghan down at Louisiana State University Health Sciences Center. Dennis was a great mentor. He was one of the original EHV researchers, did, published, many papers. What I did in his lab was I generated a virus, an EHV virus that contains beta-galactosidase, so that when we infect cells with this virus and throw on the appropriate substrate, all of the cells that are infected will stain blue. So it's an easy way for us to identify which cells are infected with EHV. And so for this experiment here, I had available a reporter virus for herpes simplex virus 1. This is the human virus that causes cold sores. So this was our control virus. Another person in the lab had generated a blue reporter virus so that any cells infected with HSV-1 would stain blue. And so what I'm showing here in these 3 panels are infection on Chinese hamster ovary cells that don't express heavy A or heavy C. If we put the virus on these cells, you see no infection. If you infect cells that now express either heavy A or heavy C, we get robust infection. Pretty much all of the cells in the plate are infected when you have one of these entry receptors expressed on these cells. So we looked at EHV, and I infected these cells with EHV, and lo and behold, I said, wow, look at this. It looks like these heavy A or heavy C may be a receptor for EHV-1. Maybe our work is done. Maybe we figured it out. However, as I always tell my virology students and students in the lab, you always want to make sure you run the appropriate controls. And so when I infected the Chinese hamster ovary cells, and remember, these guys don't express heavy A or heavy C, look what we saw. We saw that these Chinese hamster ovary cells that don't express A or C could be infected just as well, if not better, with EHV. So this made us think, right? And we deduced from this that these Chinese hamster ovary cells must have a different receptor that EHV can utilize in order to get into these cells. So it's pretty exciting, but it sent us on a journey to try to figure out what this entry receptor is. The other thing we did was at that time was to look at glycoprotein D to see, we were pretty sure that GD would be required for entry into these cells, but we wanted to test that. And so Miller-Wally was gracious enough to send us a GD knockout virus. And so when we took this virus that doesn't express glycoprotein D, and we infected Chinese hamster ovary cells or rabbit kidney cells, which are routinely used in the lab because they're easily infected with EHV, we saw little to no infection, telling us that GD is
Dr. Frampton, sorry,
yeah.
Real quick. I know we said all questions at the end of the
Sure.
video or
Right.
thing, but I was wanting to stop and ask you a question as you're talking about
Sure.
the different cells, and a question that we had and maybe help when we're going forward is what other species would what other species use cells in rabbits or rats or what other species would replicate a horse? And I'll let you go on.
I get you, Jamie. That's a great question. And you'll see that we and others have shown that in the lab, in tissue culture, it's very hard to find cells that can't be infected with EHV, regardless of the species, right? So we have monkey cells, rabbit cells, hamster cells, murine cells. We've got a ton of human cells. We've got a ton of different cells that are readily infected with EHV, right? And we'll talk about, why that may be. And it's real interesting because in the lab, we can infect all these different cells from different species, but you have a more tightly restricted host range, of course, in nature, right, at the organismal level, right, where EHV is pretty tightly associated just with equines.
Okay, so again, sorry for interrupting.
No, that's fine. That's a good question, and that's actually going to be important going forward as I talk about, the difficulty we had in actually finding cells that couldn't be infected with EHV.
Okay,
cool. All right, so we see that the GD, if you knock it out of the virus can't get into cells. So that tells us that glycoprotein D is absolutely essential for infection. When we restore glycoprotein D to this knockout virus, so that now the virus expresses this glycoprotein, now we see that we get infection, okay? All right, so the big question we had was, what receptors does EHV use to gain entry into cells? And then how do we go about finding these receptors? And as I was telling Jamie earlier, I've been fortunate here at UNC Wilmington to involve a lot of undergrads as well as graduate students in the lab to carry out this research. So here's some here and here, getting their work done. And in particular for this project, this was led by Brian Kurtz. He was one of these non-traditional students I had in my freshman cell biology class. Brian came to me after class one day and said, is there anything I can do in the lab, to help you with your research? And I said, yeah, there is, but it's going to be pretty intense. So if you're up for it, I'll put you to work. And he said, sure. And he did a great job and actually became the first author on our paper where we identified this entry receptor. So I wanted to give Brian some credit. So what we decided to do was to use an equine macrophage cDNA library. And so basically what this means is that we have a cell line where we're able to take all of the expressed genes in this cell line and make a library out of them. So we have every single gene that's expressed in this cell available. And the idea is to try to figure out which of those expressed genes is actually the receptor for the virus, okay? And this was thankfully a library that was already generated by a colleague of mine, Ron Montalero, who does a lot of work on equine infectious anemia virus. And fortunately, when I was at Pitt, he was a floor below me and I was reading some of his papers and said, well, I think I could use that library for our work, and went down and talked to Ron. He said he was very gracious. I said, sure, take what you need. And so thankfully, I got that library, and I'll show you how we use that library to isolate this receptor. All right, so don't pay too much attention. I always tell my students in my virology class, this is basically the methods, actually how we identify this receptor. But I'll skip that and boil it down to this slide here in terms of our strategy. And so I told you about these equine macrophages. We know that these cells are susceptible to infection with EHV, so they can be infected. So we know that these cells must encode a receptor for the virus, right? And so by making a library of all of the genes from this macrophage cell line, one of those genes is going to encode for the receptor that EHV uses to enter cells. So those were our susceptible cells. And what we needed to make this work is a cell line that's resistant to EHV. And as I just mentioned, it was very hard for us to find a cell line or a cell type that couldn't be infected with EHV. And we searched high and wide. And I was finally at a meeting years ago, and I was at a conference at Penn State, and I was talking to Gary Cohen and Roz Eisenberg, who've done a lot of work with other alpha herpes viruses. And they had mentioned to me that they had this really strange mouse skin cancer cell line that they had started to work with in their lab called B78H1 that was highly resistant to alpha herpes viruses that they were trying to infect cells with. So I said, that's interesting. You mind sending some of those cells to me here down in Wilmington? They said, sure. And so I brought them back to the lab. And when we put EHV onto these cells, we found that these cells were highly resistant to EHV. And that was great because the idea here was to take all of these cDNAs, all of these genes, again, one of them encoding for the receptor. We didn't know which one that would be, but the idea would be to take these expressed genes from the susceptible cells and one by one basically transfer those genes into these resistant cells until we were able to convert these resistant cells to susceptible cells, right? So the idea being when we transferred the cDNA that encodes for the EHV entry receptor from these cells into these cells now would be able to be infected with EHV. So that was a lot of work, right? We had Brian went through tons of 96-well plates, screening these cDNAs until we could convert a formerly resistant set of cells to something that looked like this. Okay, so this is what we would look for when we're screening the cDNA library. We would look to see when we get infection of these formerly resistant cells, right? And this would indicate to us that these cells now must be expressing a receptor that they gained from the macrophage cells. All right. So we got that. We got some hits, and then we went ahead and we isolated that cDNA from these infected cells, and we sequenced that gene, and what we found was Equus MHC class 1. So this was the receptor that we identified from the equine macrophage cell line that enabled EHV entry into these B78 murine melanoma cells. And so this was where another grad student of mine, actually my first grad student here at UNC Wilmington, Lauren Singletary, started to work on this project. And I tasked Lauren with verifying that this equine MHC class 1 receptor, here's a diagram of it here, actually is a bona fide receptor for the virus. We're pretty sure, but just like with anything else, before you publish, you want to make darn sure that the receptor that you've identified is indeed a true receptor for the virus. So the first thing that I had Lauren do was to clone the receptor into a plasmid, and then to take that MHC1 plasmid and to transfect these B78 cells. Remember, these are the cells that are resistant to EHV. However, when Lauren put MHC into these cells and then stained with an antibody to identify MHC1, this is what we get. So she was able to make a stable cell line, these B78 cells, all of them now expressing this MHC1 receptor. So now we've got our negative cell line here, and we've got our positive cell line here, all of these guys expressing MHC1. These guys are resistant to EHV. These guys should be now susceptible to EHV-1 because they all express MHC1. So she generated this cell line, and then to further test whether or not the receptor, the MHC receptor is actually used by EHV, she designed this really cool experiment using antibodies to block MHC1 with the idea that if you block the receptor, you're going to block infection. And we got some antibody from Doug Anchek, a collaborator at Cornell. He was nice enough to send us some of his anti-MHC antibodies. And the setup here is shown on this slide. We've got susceptible cells on the left and right. These are susceptible cells. Here's a depiction of what would be the receptor MHC1 on the surface of the cells. And so if you look at the left, if you were to just put EHV onto these cells, EHV is going to bind to the MHC receptor, and then EHV is going to make its way inside of the cell and replicate and make more copies of itself. Now, if MHC1, this receptor, is actually used by EHV to get inside of the cell, you should be able to use these anti-MHC antibodies to block all of the receptors that are available on the surface. So if you use enough antibody to coat the surface of the cell to bind to all the available MHC receptors, you would hypothesize now that the virus should not be able to enter the cell because it can't interact with its receptor because the receptors are being blocked by the antibodies. Okay, so this is a really good test for specificity of EHV interacting with MHC class 1. And so she ran that experiment. Here's the actual data from the paper. And what you see on the x-axis is dilutions of the anti-MHC antibody. So if you have highly concentrated, the more antibody you have here on the left, you get no infection, right? And as you dilute the antibody down, it's not until you dilute down to 1 to 64 until you start to see some infection of these cells. So this indicated that the antibody that can block MHC1 and prevent EHV infection. And then finally, I'll just show you this last slide here for this part of the talk. We identified the MHC as required for infection, but we had a question about whether MHC1 is involved for what we call cell-to-cell spread. So once a cell is infected with EHV, is MHC1 needed for that infected cell to pass that virus to a neighboring cell? So for this, we had infected MHC positive cells with another virus that we had in the lab that expresses green fluorescent protein. So we infect cells for 2 hours, and then we resuspended those infected cells and then mixed them with either MHC positive or MHC negative cells. And the idea was to see if MHC is required, then we should only see cell-to-cell spread on MHC positive cells and not on these MHC negative cells. So shown here is what happens when we took and mixed the infected cells with MHC negative cells. We see that we don't see any cell-to-cell spread. We just get this one single cell that's infected. However, if you take the infected cell and then you mix it with MHC positive cells, this is what you get. So vastly different. You now see this spread from one cell to another, forming this, what we call a plaque within the cell monolayer. So this told us that not only is MHC needed for infection of cells, but also is involved in what we call cell-to-cell spread of the virus. So just to recap for this part of the talk, we knew at the time that EHV could stick to the cell surface through an interaction with GB and GC, but we didn't know what the entry receptor was for the virus. We knew that EHV could get into the cell via either direct fusion or endocytosis and traveled along microtubules to the nucleus. And then what we learned from our study was that indeed glycoprotein D is essential for entry to occur. We also showed that MHC1 is an entry receptor for the virus. And we were on our way to looking at GD interacting with MHC1. We were doing some of those, running some of those experiments, but a group in Japan led by Sasaki actually beat us to the punch and published first. And they actually showed that GD physically interacts with MHC1. And after this interaction, the virus gains entry into cells. And so we now identified this receptor and how the virus binds to it. So hopefully, we can put this knowledge to use and maybe come up with some antivirals that could block this interaction between the virus and the receptor. So that work is still ongoing. All right, so I want to switch gears a little bit here and talk about the current Grayson funded work that we're doing. And we're kind of going to flip the script a little bit here. And for this research, what we're doing is we're looking at the immune response that's triggered after EHV infection. And then whether there's any interventions that we can come up with to try to slow down or to inhibit this pro-inflammatory response that we see. And so this work was started by Stephanie Johnstone and Kate Arnett. Stephanie just finished her PhD at North Carolina State a couple weeks ago, so very proud of Stephanie. Kate went on from the lab to become a nurse, and so she's on the front lines right now of the COVID pandemic. But both Stephanie and Kate did most of the work for this initial paper. And what we looked at was EHV-induced immunopathology. It's been well described in the literature that a lot of the pathology that's seen post-EHV infection is driven by the immune response to the infection and the overexuberant immune response and inflammatory response. And so, for example, equine herpes virus myeloencephalopathy has been shown by multiple groups to be a disease primarily caused by severe inflammation within the central nervous system. So the endothelial cells within the central nervous system are infected after the cell-associated viremia, and you get the production of these pro-inflammatory cytokines that then go on and recruit immune cells to the site of infection, and you get this severe immunopathology. Some of the inflammatory chemokines that I'm going to talk about here in a minute that we're currently investigating include CXCL9, 10, and not shown on here, CXCL11. These guys, when they're expressed, they are able to recruit multiple immune cells to the site of infection. And if you get overexpression of these pro-inflammatory chemokines, you get hyperinflammation and immunopathology. So a little bit of expression of these guys is a good thing, but too much expression is damaging to the horse. And I just want to show this slide here. This is work that we did years ago in a mouse model of EHV infection, where we were looking at whether or not we could replicate the neurologic disease in mice. And we were looking at different strains of virus to see if some strains were more neurologic than others. And so we showed with a virus that, so here is Kentucky A, a vaccine strain. And when we infected mice with this vaccine strain, we get some encephalitis in the brain tissue, but no severe encephalitis or meningoencephalitis. However, if we put glycoprotein I and E back into the strain, we now see a more severe meningoencephalitis with recruitment of immune cells into the brain tissue. And so this led us to search for mediators of immune recruitment and how they participate in immunopathology that's triggered by EHV. So we had two hypotheses at the time. Number one was that neurologic strains of EHV replicate more efficiently than non-neurologic strains in equine endothelial cells. And so as you probably know, there are certain strains of EHV that are labeled as neurologic if they have a particular mutation in the viral DNA polymerase, and other strains that are identified as non-neurologic if they don't contain that mutation. So one hypothesis we had at the time was that the neurologic strains may cause more severe disease simply because they can replicate better in certain cell types. So that's what we wanted to test. And then a separate hypothesis we had was that these neurologic strains can induce a more severe pro-inflammatory response. That is, we hypothesized that these neurologic strains would be able to induce higher expression of these pro-inflammatory chemokines like CXCL9, 10, and 11. So we carried out that work, and when we looked at endothelial cells and the amount of virus that was produced in endothelial cells that were infected with non-neurologic strains here on the left versus neurologic strains here on the right, we basically saw no difference in the ability of these strains of EHV to infect endothelial cells. So that kind of shot down that hypothesis, right? We didn't see neurologic strains of the virus being better able to infect these cells versus non-neurologic. So we then wanted to look at the amount of the cytokines, the pro-inflammatory cytokines that were induced after infection with neurologic and non-neurologic strains. And so the setup is shown here. We'd infect these endothelial cells with EHV for 10 hours and then isolate total RNA and run through this process where we do real-time quantitative PCR to measure the amount of pro-inflammatory cytokines that are triggered after infection with EHV. And when we did this, a few genes popped out at us. So highlighted in yellow here are the pro-inflammatory chemokines, CXCL9, 10, and 11. All of these were significantly upregulated compared to mock infected cells. And importantly, we basically saw no significant difference in the expression of these pro-inflammatory chemokines after infection with non-neurologic EHV or neurologic strains of EHV. So that shot down hypothesis number 2, right? We originally thought that the neurologic strains would trigger more production of these pro-inflammatory chemokines versus these non-neurologic strains, but we didn't see that in our data. So that led us to wonder what's going on, and we and other groups are still trying to tease out why these neurologic strains cause more disease than non-neurologic. Now, I don't want everybody to get the impression that the non-neurologic strains never cause disease. It's just that about 80% of the time when you get equine herpes virus myeloencephalopathy, it's caused by a neurologic strain. But there have been cases where a horse has EHM, and then when they type the strain, they see that it's a non-neurologic. Okay, so we saw that these chemokines were highly upregulated after infection. So we wrote a grant, and Grayson funded this work. And the idea is, can we find any drugs that can reduce the expression of these pro-inflammatory chemokines so that we can possibly reduce the inflammation that contributes to the neurologic disease, right? Can any of these drugs tamp down this inflammatory response? So I'm not going to read through all this, but basically we know that these three chemokines are highly upregulated, and these guys are also upregulated after infection with a variety of other viruses, including coronavirus, including SARS-CoV-2. So this pro-inflammatory response, including CXCL9, 10, and 11, have been shown to contribute to inflammation and neuropathology with a host of other viral diseases or infections. All right. So we wanted to investigate some drugs that are already used in horses to reduce inflammation caused by various triggers. So multiple inflammatory drugs, including flunixameglumine, ketoprofen, phenylbutazone, dexamethasone, firecoxib, have all been used to reduce inflammation in horses. However, there's very limited data on the efficacy of these compounds in reducing inflammation induced by EHV. So we wanted to test these guys to see if any of these guys could reduce the inflammation that's triggered by EHV infection. And so we were looking at those, and then we're also looking at another class of inhibitor called the Janus kinase inhibitor, or JAK inhibitor. These are prescribed in humans. One name brand is Xeljanz, and they've been used to depress inflammation. And so we investigated this guy, and we're also investigating this other inhibitor that we previously showed inhibits EHV infection. So the hypothesis here would be that EHV yields and cell-to-cell spread would be reduced in the presence of these drugs and JAK inhibitors, and also that the EHV-induced pro-inflammatory cytokines 9, 10, and 11 would be depressed by these drugs. So here are the drugs that we used in the study, and I'll just show you how we set up these experiments, and then show you real quickly some of the data that we're currently generating to address these hypotheses. So first here is the inhibition of virus yield assays. As I mentioned at the beginning of the talk, we were displaced from our usual lab because of the hurricane, and a lot of the data I'm going to show you was what I did in the spring and summer over in Friday Hall, just running through these assays one after the other. So we were able to generate quite a bit of data looking at the ability of these drugs to block EHV infection. So real quickly, so the way we do this is we add these different drugs to cells for 24 hours. Then on day 2, we add fresh drug plus infect with EHV. And we use 2 different multiplicities of infection, 0.1 and 1. 0.1 means for every 10 cells, we add 1 virus particle, and MOI of 1 means for every cell in the monolayer, we add 1 virus particle, 1-to-1 ratio, virus to cell. We then, on day 3, harvest the total virus, and then we run what we call a plaque assay, where we make 10-fold dilutions of the virus, put it onto cells and plates, and this is what one of these titer plates looks like, right? And so we tested 2 neurologic strains, or are currently testing 2 neurologic strains. T953 is the Ohio 2003 strain that caused the outbreak in Findlay, Ohio. And T967 is another neurologic strain. And then we've got these 2 non-neurologic strains. And for each one of these, the assays were run 3 independent times in order for us to generate our data. And here's some of what the data that we've generated on the rabbit kidney epithelial cells. And so cells that were treated with the JAK inhibitor AG490 shown here, or flunixin meglumine shown here, we get a significant reduction versus cells that are treated with the vehicle alone, the DMSO. So this is basically untreated cells here, the amount of virus that's produced in cells that are treated with no drug, and then we compare that with cells that are treated with one of these drugs. And so the JAK inhibitor AG490, we got a pretty significant reduction in virus yield with that JAK inhibitor. And we also got a pretty significant drop in infection when these cells were treated with flunixin meglumine. Now, these other drugs, we didn't see much of an effect, a little bit with the ROCK inhibitor, but the dexamethasone, the firecoxib, ketoprofen, and phenylbutazone, we didn't see a reduction in infection. Here's what we see with the Ohio 2003 strain. Again, we see that the JAK inhibitor AG490 and flunixin meglumine were able to inhibit virus yield in these cells. So that was pretty cool data showing that these two guys could reduce the amount of virus that's produced in cells. If we look at the same cells, but now we're infecting with a higher amount of virus, MOI of 1, we still see that AG490 and flunixin meglumine are able to, even at a higher dose of virus, these two drugs are able to reduce infection with both the neurologic strain as well as the non-neurologic, and the neurologic strain. So that's pretty cool because the idea is that if you can reduce the amount of virus that's produced, let's say in the respiratory epithelial cells of a horse, that's going to go a long way towards reducing the amount of virus that gets into the bloodstream that can make its way to the endothelial cells in the CNS. And so anything that you can do to reduce viremia is going to be a win for the horse. We then ran some viruses on the equine endothelial cells. So it's the same experimental setup, just using the endothelial cells instead of the epithelial cells. And this is data, these data here are with the EHV at MOI of 0.1. And interestingly, on these endothelial cells, we still see nice inhibition with the JAK inhibitor AG490, shown here, here, and here, on 3 different strains of EHV. But on these endothelial cells, we don't see a similar reduction with flunixin meglumine or any of the others. So that was interesting to us, that the JAK inhibitor still works on endothelial cells, but the flunixin meglumine not so much. And then quickly, I'll just show, this is the MOI of 1. Even at MOI of 1, we still get a reduction on with the AG490 JAK inhibitor here and here. However, we didn't see a significant reduction at MOI of 1 with the Ohio strain. And so I know I'm probably running short on time, but I'll real quickly just set up what we're currently doing in terms of looking at the ability of these drugs to not only reduce virus replication and yield, but to reduce the expression of these chemokines from cells. So this is work that's currently being run by my student Jacob Kaz-Nelson, as well as Jenny Black in the lab. And they are looking at, is there anything we can do to reduce the expression of these guys? And so, for example, does the JAK inhibitor AG490, if you put it onto these cells and then infect with the virus, do we see a reduction in these chemokines? And so this is some preliminary data that was generated. And what you're seeing, what we're showing here is the amount of CXCL10 and 11, these 2 pro-inflammatory chemokines. If you have no virus, of course, you don't get very much expression. If you add the Ohio strain and in the absence of any drug, you get nice expression of this pro-inflammatory chemokine. With the control DMSO, you still get nice expression of this chemokine. However, if you treat cells with the JAK inhibitor and then infect, you get a reduction in the amount of this pro-inflammatory chemokine. And then with CXCL11, we saw a similar reduction. Here's the virus inducing this chemokine. You add the JAK inhibitor, you get a nice reduction in the expression. So this shows us that not only does the JAK inhibitor reduce the amount of virus that's produced in cells, but it's also reducing the amount of the pro-inflammatory chemokines that are expressed from these cells. So it's kind of what we think of as a win-win, right? And that's kind of shown here on this slide that kind of wraps up the whole talk. So we've shown that the JAK inhibitor and flunixin megalamine are able to reduce EHV production in epithelial cells. So you can imagine that in a horse that's treated with one of these drugs, you may reduce the amount of virus that's produced that could make its way into the bloodstream. So we inhibit infection in the respiratory tract, we reduce viremia, and we reduce the amount of virus that makes its way to these sensitive endothelial cells in the pregnant uterus or in the central nervous system. And we also reduce the amount of chemokines that are expressed from these endothelial cells. So we reduce the inflammation present in these cells. So all in all, if we can kind of hit the virus at these different places, different time points, the idea is that we're going to go a long way towards lowering the risk of neurologic disease or an abortion disease outcome. So this is some of the current work we're doing. So we're finishing up our virus yield assays with our T967 and Kentucky A strain. We're going to look at the ability of these drugs to block cell-to-cell spread. We're going to continue to look at inhibition of chemokines by compounds. We're going to see when these chemokines are expressed post-infection and try to determine which EHV proteins actually are responsible for inducing these pro-inflammatory chemokines. And with that, I'll take any questions. I thank everybody for listening and allowing me to talk about the research that we've done. I want to acknowledge Grayson Jockey Club Research Foundation for funding all of this work, as well as some of our other funding agencies, all of the lab members that actually ran all these experiments, and of course all of my collaborators who, without their help, I wouldn't be able to complete this work. So I thank you, and I'll take any questions that you have.
Thanks so much, Dr. Frampton. We can, let's see, stop it if you, okay, so yeah, we do have a few questions. Let me get to this first one. This is a pretty direct one that is going to be directed right to you. Is the classification of EHV-1 to neuropathogen and non-neuropathogen due to the mutation of polymythos gene? I don't know if I'm saying that right.
Right, yeah, yes, the way that they're classified is based on the mutation in the DNA polymerase.
Is that still valid or not is the question.
That's a great question, and kind of yes and no, I guess. So it has been shown that having that mutation allows for more replication a little bit and a little bit higher viremia, but it's not open and shut, right? It's not a black and white. So it's not as if you just have that mutation, you're neurologic, and if you don't, you're non-neurologic. There are other factors that come into play that allow for one disease outcome versus the other, right? And so one of the things that we would like to do is to sequence all of these strains to identify other factors, other viral factors that actually contribute to one outcome or the other. So there's more to it than just the DNA polymerase mutation. That plays a part, but it's not the be all and end all.
Okay, so maybe a little bit more general then is, you talked about reactivation of the virus can occur. If they're vaccinated, does the reactivation have the same symptoms? Are they still just as contagious to pass it along? How does that all work with the virus and reactivation?
Yeah, so a lot of that depends on, the time from vaccination until the horse reactivates, right? So you're going to have more protection the closer you are to the vaccination, right? And so if you're a year or two out and you haven't been vaccinated and you reactivate, you're going to potentially see, virus shedding and symptoms occurring in that horse. And that's a real issue with these vaccines, is that there have been many that have been, developed, and, some have been shown to be somewhat effective, but none of them so far have been able to show long-term immunological protection with just one or a couple of doses, right? And that's a problem where these horses have to be vaccinated continuously, right? Those horses that are on the racing circuit are going to be vaccinated with EHV sometimes, every 3 months. It's
Hmm. So the next question said, what about EHV-8?
Hmm. Yeah, I really haven't done anything with EHV-8, so I really don't have a whole lot on knowledge on that one. Most of our work has been on 1 and 4, and primarily 1.
Okay. Another person asked, are the overexpressions of CXCL9-11 a common trait in other viral infections beside EHV-1?
Yes, they are. And that was actually, so on one of those slides there I had, you look at influenza virus, dengue virus, SARS-1 and 2. There's been research done that these pro-inflammatory chemokines are a key driver of immunopathology. So anything, and flu is another big one. When you think about the 1918 pandemic, the Spanish flu and bird flu, these strains have been shown to trigger a real severe hyperinflammatory response. And so anything that can be done to kind of tamp that down is going to be helpful. So it's not just the virus itself. A lot of people think of viruses as destroying cells and causing all the disease on their own. But there's more to it than that. A lot of the time where you see deaths, severe illness and deaths associated after infection is due to the overexuberant immune response that you'll see to these viruses. Of course, you need some of an immune response to clear the virus and recover, but in many cases, you get too much of an inflammatory response that's damaging.
Yeah, so another question, and we've been reading a lot about with the COVID vaccination and the messenger RNA.
Mhm.
We actually have a project here at Grayson, which is trying messenger RNA with rhodococcus.
Yeah, I was just talking about that very thing right before with my grad student Kaz, right before coming on this webinar. I was telling him that last night, I was thinking about this talk and, watching all the news with COVID and of course the messenger RNA vaccines. And I was thinking that would be really cool to try in the horse, right? We've tried subunit vaccines with glycoprotein D, different glycoproteins, but the protein itself is going to induce an antibody response, what we call a humoral response, but it's not going to develop, you're not going to trigger a cell-mediated immune response, right? You're not going to get good CD4 and CD8 cytotoxic T cells generated based off of that type of protein vaccine. But with a messenger RNA vaccine, one of the real values of the messenger RNA vaccine is that they have been shown to trigger both arms of the adaptive immune response. So you're going to get antibodies against the protein that's produced from the messenger RNA, like the spike protein from SARS-CoV-2. But also your cells are going to take that messenger RNA up and they're going to make the viral protein in your cells. And so to the organism that's been vaccinated, that messenger RNA that's being generated is going to be seen as being foreign, and they're going to think that they're actively infected with virus. And so what that's going to do is that's going to allow those cells to display that viral protein on their surface, and that will trigger cytotoxic T cells to come in and kill those cells and activate that CD8 cell-mediated immune response. So you get both arms. And there's been plenty of work done with EHV to show that you need both arms of adaptive immunity to have a better protection, better immunological protection. So I really think, and maybe that'd be the next grant, right? I really think that investigating messenger RNA vaccines, maybe we've got the 12 glycoproteins, you can make messenger RNA for each of those 12 and see what kind of immune response you get if you vaccinate using a messenger RNA.
Yeah, and so for those that asking, we are recording this and we'll post it to YouTube and send out a tweet and some social media messages about when it's posted. But another question is, somebody wants to know if there's any experiment that uses the JAK3 inhibitors in horses.
Not that I've come across yet. So that would be, something that, we're getting some good results in tissue culture. The next step would be to test these for safety and efficacy in a small animal model. And we've developed small animal mouse models, so we could investigate safety and efficacy in a small animal model, and then it would go up to horses. But to my knowledge, nobody's looked at those yet. I think we're the first actually to investigate these JAK inhibitors against EHV.
Well, I have a question. I just have one. It's since we can replicate EHV-1 in many different animals and cells and species, how much of that information or how much of those other studies are transferable over, and how does it further our studies in the equine version?
Yeah, so I think it's like, so you're talking about, the ability of EHV to infect multiple different cell
Yeah,
types.
you said small animal trials, and how much does that further your study and help you guys kind of jumpstart the equine research when you're able?
Yeah, I think it has a lot of value. Again, most of the work that we do here is all tissue culture, but we have done, and I showed some of the slides that we've done in the mouse, and of course, no model is perfect, but I do think that we can learn a lot in terms of pathogenesis of the virus using a small animal model that it would be useful and give us information that's important for the equine. Again, it's not important, but in terms of investigating, for example, can we knock down the amount of virus that gets into the bloodstream in an animal, in a small animal, we would, I'd be fairly, confident that, any drug that would knock down viremia in a mouse model should be able to knock down similarly in an equine. So I think there's a lot in terms of the pathogenesis, pathology that ensues after infection that we can learn in the small animal model that would be applicable to the equine.
That's great, because I know we have some other research in other equine diseases that we just can't replicate in the lab, or we can't replicate with other animal models. So I'm sure that those researchers are a little jealous that you're able to do this in tissue and replicate it across other kind of species.
Right, and like one thing that we want to follow up on that kind of can't do everything, I guess, but we do know that there are certain glycoproteins that look to be important for allowing the virus to enter into the central nervous system. So, for example, the Kentucky A strain is a vaccine strain that doesn't express a couple glycoproteins, glycoprotein I and glycoprotein E. And so I put those two glycoproteins back into that vaccine strain to make a recombinant virus. And when those two glycoproteins were reintroduced into that vaccine strain, they were now able to get to the brain and cause a more severe disease and to recruit immune cells. And so that shows us right there that those two glycoproteins play a role in the neuropathogenesis of the virus. So we might want to develop drugs that can, inhibit glycoprotein I and E.
Yeah, well, I don't see any other questions, but I just wanted to say thank you so much for taking your time, and thank you for rescheduling.
Yeah,
a very important problem that there's not an immune horse out there for this virus, and as we all know, when it gets in a horse show facility or a barn or a backside of the racetrack, it renders commerce and horses.
David, that's one more thing if I could add in terms of quelling outbreaks. I think if we show that some of these anti-inflammatory drugs or JAK inhibitors, we can actually use them prophylactically, right? Possibly, right? So if you have an outbreak and you identify EHV early, you can go ahead and give all of your horses these anti-inflammatories and JAK inhibitors to basically better protect them. Doesn't mean they wouldn't get infected, but right, but maybe you reduce that viremia and you reduce the chance or the risk that those horses could get those severe disease.
And I would imagine, like all of this stuff, that only helps when you have early detection and allows you to act with your veterinarian team a little bit faster. So
exactly.
I had somebody said thank you for the presentation. So it's been very useful, and like we said, we'll post it up on YouTube this afternoon, and we'll put a note out to everybody when it's up. And Dr. Frampton, thank you so much for your time today, and more importantly, thanks for your work and everything that you do for horses.
Okay, thank you so much, Jamie. Thanks to Grayson. I appreciate it.
Yeah, thanks, guys. Everybody have a great day out there. Stay safe.
Okay, thanks.